Welcome back to the deep dive. Today we're plunging into one of the most fundamental yet astonishingly complex processes in human physiology,
Mm,
how our nerves command our muscles to contract.
mm.
It's the basis of every movement, really, from blinking to running a marathon, and we're going to unpack it to a level that, well, really aims for mastering the details,
Mhm,
like the
mm.
elite professionals in the field do.
Exactly. And we've got some really solid material here to guide us through the mechanics of the neuromuscular junction and this thing called excitation-contraction coupling.
Right.
Our mission basically is to go deep. Look at the structures, the chemical signals, the electrical events, the precise coordination needed to make movement possible. We'll try and cover everything in the sources, really focus on understanding every concept.
Think of it like getting an expert-level blueprint of your body's own motion control system. We're exploring that critical synapse where nerve meets muscle, the intricate dance of molecules, and uh, how a signal on the muscle surface triggers action deep inside the cell.
And we'll definitely look at what happens when this incredibly precise system hits a snag, whether it's disease or maybe targeted drugs. So, yeah, let's dive in. Where does this command chain begin? Right at the muscle itself.
Okay, let's unpack this. So it starts with the connection point. The skeletal muscle fibers,
Yeah.
the ones under voluntary control, they get their instructions from these large myelinated nerve fibers.
Mhm.
And these originate way up in the spinal cord from the large motor neurons there.
Right. And these nerve fibers, they aren't just one-to-one partners with muscle fibers, are they?
No, exactly. A single nerve fiber, once it enters the muscle, it branches out, which means it can stimulate multiple muscle fibers all at once.
And that number can vary quite a bit, right?
Yeah, the range is pretty wide. Could be just a few, maybe three, or it could be like several hundred muscle fibers. It depends on what the muscle is designed to do.
So one nerve signal can orchestrate a whole group.
Okay.
And where does that nerve fiber actually, connect to each muscle fiber?
Each branch forms a junction, typically right near the middle of the muscle fiber. That specific spot is the neuromuscular junction.
Ah, the NMJ.
Yes. And what's interesting is the signal, the electrical signal that starts in the muscle fiber. It actually travels out in both directions along the fiber from that central point.
That makes sense, yeah. Spreads it out efficiently. And is it common for a muscle fiber to get signals from multiple nerves or multiple spots?
Actually, no, it's quite specific. The sources say, with very few exceptions, maybe like 2%, each skeletal muscle fiber gets input from only one neuromuscular junction. It's like a dedicated phone line.
One input per fiber. Got it. Okay, let's zoom in on that junction structure. It's often called the motor end plate. But the nerve and muscle, they don't actually merge or fuse, do they?
No, they don't. The nerve fiber endings, they kind of tuck themselves into the surface of the muscle fiber. They invaginate. But technically, they're still outside the muscle fiber's own plasma membrane.
Okay, so nestled in, but separate. And this whole area, it's insulated.
Yeah, the whole motor end plate structure, the nerve terminal tucked in there, it's covered by Schwann cells.
Yeah.
Provides insulation.
Right. So we have this nerve ending nestled into the muscle surface. What's the gap like between them?
Well, that depression in the muscle membrane, where the nerve ending sits, that's sometimes called the synaptic gutter or trough.
Okay.
The actual space separating the nerve membrane from the muscle membrane, it's a synaptic space or synaptic cleft. And it's incredibly narrow, like 20 to 30 nanometers wide,
Tiny, like virus-sized gap.
pretty much.
Huh.
And the muscle membrane on its side of that gap, it's not smooth.
No, not at all. At the bottom of that synaptic gutter, the muscle membrane has loads of smaller folds. These are called subneural clefts.
And why the folds?
Surface area.
Hmm.
They massively increase the surface area of the muscle membrane that's available to interact with the chemical messenger coming across the gap. More area means more efficient signaling. It's crucial.
Okay, scene set. Now, what's going on inside that nerve terminal to get the signal ready? What's it packing?
Right. Inside the terminal, it's basically a little factory. It's packed with mitochondria, the powerhouses.
ATP.
Exactly. They supply the ATP needed to synthesize the main neurotransmitter here, which is acetylcholine, often just called echo.
And it makes the acetylcholine right there.
Yep, synthesized right in the cytoplasm of the nerve terminal.
Okay, so it makes ECHI. Is it just floating around then?
No, it's immediately snapped up and packed into tiny storage bags. These are the synaptic vesicles.
Ah, the vesicle.
And there are a lot of them. A single end plate terminal might have around 300,000 of these vesicles ready to go, each one crammed with acetylcholine, like 10,000 molecules per vesicle.
Wow, that's a huge stockpile. ready for action. And what about the synaptic space, that tiny gap? Is it just empty fluid?
Nope, not empty. It contains large quantities of a crucial enzyme called acetylcholinesterase.
The cleanup crew.
Exactly. Its whole job is to break down acetylcholine extremely rapidly, like milliseconds after it's released. This quick cleanup is absolutely vital for controlling muscle activity, preventing continuous firing.
Okay, so the nerve terminal is loaded with vesicles. A nerve impulse, an action potential arrives. How does that electrical signal actually trigger the release of the chemical message?
Right, so the action potential sweeps down the nerve fiber and hits the terminal. This kicks off the release process. On average, about 125 vesicles worth of acetylcholine get dumped into the synaptic space with each single nerve impulse.
125 vesicles per impulse. How does the electricity make the vesicles release?
It's all about calcium. On the inside surface of that nerve terminal membrane, there are these structures called linear dense bars.
Dense bars. Okay.
And right next to these bars, embedded in the membrane, are special protein channels. These are voltage-gated calcium channels.
Voltage-gated, so they open when the electrical potential changes, like during an action potential.
Exactly. When the action potential arrives and depolarizes the nerve terminal membrane, these calcium channels pop open.
And where does the calcium come from? Is it stored inside?
No, it comes from outside. Calcium ions are much more concentrated in the fluid within the synaptic space compared to inside the nerve terminal. So when those channels open, calcium ions flood into the nerve terminal, flowing down their concentration gradient.
So calcium rushing in is the trigger.
That's the key signal, yes. Inside the terminal, this influx of calcium is thought to activate an enzyme pathway. It involves something called K2 plus calcineurin-calmodulin-dependent protein kinase.
Yeah.
This kinase then adds a phosphate group, phosphorylates these proteins called synapsins.
Synapsins. What do they do normally?
Normally, synapsins act like little tethers. They anchor the acetylcholine vesicles to the cytoskeleton, the internal scaffolding of the nerve terminal, keeping them parked.
Ah, so phosphorylating them cuts the ropes.
Precisely. Phosphorylation frees the vesicles from their anchors. This allows them to move towards the membrane, specifically to areas called active zones, right near those dense bars we mentioned. The vesicles then dock at specific release sites on the membrane, fuse with it, and spill their acetylcholine contents into the synaptic space. It's a process called exocytosis.
So the electrical signal opens calcium channels, calcium comes in, activates a kinase, kinase frees the vesicles, vesicles move, fuse, and release He.
You got it. It's a beautiful cascade. Some finer details are still being worked out, but the core idea is solid. Calcium entry near those dense bars is the direct stimulus for acetylcholine release.
Okay, signal sent. Acetylcholine floods that tiny synaptic cleft. What's happening on the muscle fiber's side? How does it hear this message?
The muscle fiber membrane is listening very carefully. It's studded with lots of acetylcholine receptors, and also, importantly, lots of voltage-gated sodium channels nearby.
And where exactly are these acetylcholine receptors located? Are they spread out?
No, they're highly concentrated. These acetylcholine-gated ion channels, the receptors themselves, are located almost entirely near the openings, the mouths of those subneural clefts.
Those folds we talked about.
Exactly, which makes perfect sense, right? They're right where the acetylcholine is being released from the nerve terminal just across the gap. The voltage-gated sodium channels are also lining these clefts, ready to act next.
Smart design. Tell me about the receptor protein itself. Is it simple?
Oh no, it's a pretty hefty, complex protein embedded in the muscle membrane. Molecular weight around 275,000. In adults, it's made of 5 distinct protein subunits. 2 called alpha, 1 beta, 1 delta, and 1 epsilon.
And those alpha subunits are key.
Very key, because they are the parts that actually have the binding sites for acetylcholine.
And these 5 subunits together form a channel.
Yes, they arrange themselves to form a tube-like channel that goes right through the muscle membrane. But normally, when no acetylcholine is around, that channel is closed, constricted.
Okay, here's the crucial bit. How does acetylcholine open this channel?
It takes 2. For this channel to open, 2 molecules of acetylcholine must bind simultaneously, 1 to each of those 2 alpha subunits on the receptor.
Both at the same time.
Yes. That double binding causes a conformational change, basically. The protein twists slightly, and that physical change opens up the central pore of the channel.
And when it's open, what can get through?
The open channel pore is about 0.65 nanometers wide. That's big enough for important positive ions, cations like sodium, Na+, potassium, K+, and even calcium, C at 2+, to slip through fairly easily.
What about negative ions like chloride?
Nope. Negative ions, anions like chloride, are repelled. There are negative charges lining the mouth of the channel pore that effectively block them out.
So only positive ions flow. But the sources really emphasize sodium. Why does so much more sodium flow through than potassium or calcium?
Two main reasons, both pushing sodium inwards.
Hmm.
First, concentration gradients. Sodium is way more concentrated outside the muscle cell than inside. Potassium is the opposite, high inside, low outside. So just chemically, sodium wants in, potassium wants out.
Okay, chemical push. What's the second reason?
It's electrical. The inside of a resting muscle fiber membrane is very negative, around negative 80 to negative 90 millivolts. This strong negative charge inside acts like a powerful magnet for positively charged sodium ions, pulling them into the fiber.
Mm.
At the same time, that negative charge inside opposes the exit of positively charged potassium ions, even though their chemical gradient wants them to leave.
So the electrical pull for sodium is huge, and it hinders potassium leaving.
Exactly. The net result is that the dominant effect by far of opening these acetylcholine-gated channels is a massive influx of positively charged sodium ions into the muscle fiber.
And all that positive charge rushing in must change the voltage inside the muscle fiber, right there at the junction.
It does, immediately. This localized influx of positive sodium ions creates a sudden, local positive shift in the membrane potential right there at the motor end plate. This local potential change is called the end plate potential.
The EPP. And this is the spark that lights the fuse for the muscle contraction.
It's the crucial first step on the muscle side. Normally, this end plate potential is pretty substantial. It typically increases the membrane potential inside the muscle fiber by, say, 50 to 75 millivolts in the positive direction.
Okay.
And that depolarization is usually more than enough to reach the threshold needed to trigger the opening of those nearby voltage-gated sodium channels, which are also clustered in the subneural clefts.
So the acetylcholine opens its channels, sodium rushes in creating the end plate potential, and if that potential change is big enough,
It hits the threshold for the voltage-gated sodium channels. They then fly open, causing an even bigger self-sustaining rush of sodium ions into the cell. That's the regenerative part.
and that's the muscle action potential.
That's the full-blown muscle fiber action potential. It then propagates, spreads like a wave, along the entire length of the muscle fiber membrane, carrying the contract signal.
Signals successfully jump the gap. But, like you said before, that acetylcholine signal in the cleft needs to be shut off fast, right? Otherwise, the muscle would just stay contracted.
Absolutely critical. Rapid removal is key. If acetylcholine just hung around in the cleft, it would keep binding to receptors, keeping the muscle membrane depolarized, or causing it to fire over and over again. You'd get uncontrolled muscle spasms or paralysis.
And that's where acetylcholinesterase comes back in.
Yes, the enzyme we mentioned earlier, it's the rapid deactivator. Acetylcholine gets quickly hydrolyzed, basically chopped in half by acetylcholinesterase. Most of this enzyme is anchored in that spongy connective tissue filling the synaptic space.
So it cleans it up right there.
Exactly. A tiny bit of acetylcholine might also just diffuse away out of the cleft, but the enzyme does the heavy lifting.
And how fast is rapid?
Extremely fast. Acetylcholine usually only exists in the synaptic space for a few milliseconds, maximum. Just long enough to bind, open the channels, create that end plate potential, and initiate the muscle action potential. Then, poof, it's gone.
So the muscle can relax and be ready for the next signal.
Precisely. This rapid on-off switching is fundamental for allowing controlled cycles of muscle contraction and relaxation. Thinking about that end plate potential again, that 50 to 75 millivolt positive change, you said it's usually more than enough to trigger the action potential. How much is actually needed? The threshold, the minimum depolarization needed to reliably open those voltage-gated sodium channels and fire a muscle action potential, is typically only around a 20 to 30 millivolt positive change from the resting potential.
So 50 to 75 is way over that. That sounds like a safety margin.
Exactly what it is. It's called the safety factor. Under normal circumstances, each nerve impulse generates an end plate potential that's about 3 times stronger than the bare minimum required to stimulate the muscle fiber.
So the normal neuromuscular junction is built to be highly reliable. Less chance of a signal getting dropped.
Very reliable, yes. It has a high safety factor for transmission.
But is it foolproof? Can this system get overwhelmed?
It's not limitless. If you stimulate the nerve fiber at incredibly high rates, the source mentions over 100 times per second for several minutes, which is pretty extreme, the nerve terminal can actually start to run low on readily available acetylcholine vesicles.
It can't keep up with the demand.
Right. The synthesis and packaging process might not keep pace with such rapid release. If the number of vesicles released per impulse drops significantly,
Then the end plate potential gets smaller.
exactly, it might fall below that critical threshold needed to trigger the muscle action potential. The signal fails to transmit. This is known as fatigue of the neuromuscular junction.
Is this the main cause of muscle fatigue when we exercise hard?
It can contribute potentially under very extreme conditions. It's similar to synaptic fatigue seen elsewhere in the nervous system, but honestly, under normal physiological conditions, during everyday activities, significant neuromuscular junction fatigue is rare. That high safety factor usually prevents it. It really only becomes a potential issue during the most exhausting types of muscle activity.
Okay, let's piece together the whole story of acetylcholine at this junction. It sounds like a really efficient cycle.
It absolutely is, a constant turnover. So step one, the empty vesicles themselves, those little 40-nanometer bubbles, they're actually made way back in the cell body of the motor neuron in the spinal cord by the Golgi apparatus.
And ship down the axon.
Yeah, transported all the way down to the nerve terminal by this process called the axoplasm streaming, kind of like a conveyor belt inside the nerve fiber. And about 300,000 of them collect at a single end plate.
Okay, empty vesicles arrive, then they get filled.
Right. Acetylcholine is synthesized in the cytoplasm of the terminal. As soon as it's made, it's actively pumped into these vesicles and stored there at that super high concentration, 10,000 molecules per vesicle.
Then the action potential arrives.
Yep. Triggers the voltage-gated calcium channels to open. Calcium rushes in about a 100-fold increase inside the terminal.
And that triggers release.
A massive 10,000-fold increase in the rate of vesicle fusion with the terminal membrane. About 125 vesicles rupture per action potential, releasing their HE payload via exocytosis into the synaptic space.
Then the quick breakdown.
Within milliseconds, 5 to 10 milliseconds for the whole sequence, maybe acetylcholinesterase splits ALT into acetate and choline. And here's the recycling bit. The choline is actively pumped back into the nerve terminal.
To make more acetylcholine.
Exactly. It's immediately reused to synthesize new acetylcholine molecules. A very neat closed loop for the chemical itself.
But what about the vesicle membrane that fused with the terminal membrane? Does the terminal just get bigger and bigger?
No, that membrane needs to be retrieved. Otherwise, the terminal would run out of membrane and vesicles quickly. Remember, those 300,000 vesicles are only good for maybe a few thousand nerve impulses.
So how does it get the membrane back to make new vesicles?
It's a process called endocytosis. Within seconds after the vesicles have fused and released their contents, little indentations called coated pits appear on the nerve terminal membrane. These are formed by contractile proteins, especially one called clathrin, that gather on the inside surface of the membrane where the vesicles fused.
Coated pits, and they pinch off.
Exactly. Within about 20 seconds, these clathrin coats and associated proteins contract, causing the pits to deepen and eventually pinch off from the surface membrane, forming brand new empty vesicles back inside the terminal.
Whoa.
And then, within a few more seconds, acetylcholine is transported into these newly recycled vesicles, getting them ready for the next round of release. It's a continuous, efficient recycling process for both the messenger and its container.
That whole system, the precise release, the specific receptors, the rapid cleanup, the recycling, it's incredibly intricate. It makes sense that messing with any single part could have big effects.
Oh, absolutely. The neuromuscular junction is a prime target for a lot of things. Drugs, toxins, even the body's own immune system, sometimes precisely because disrupting it has such immediate and obvious effects on movement.
What are some ways drugs target this junction?
Well, one group of drugs basically mimics acetylcholine, things like methacholine, carbachol, and even nicotine. They bind to the acetylcholine receptors on the muscle fiber and open the ion channels, just like ACET does.
But what's the difference then?
The key difference is they aren't broken down quickly, or sometimes at all, by acetylcholinesterase. So instead of a brief millisecond pulse, their effect lasts for minutes or even hours.
And what does that do to the muscle? A constant on signal.
It causes a persistent depolarization right at the motor end plate. This initially triggers action potentials, but the sustained depolarization can actually make the nearby voltage-gated sodium channels refractory, unable to fire again immediately. However, leakage currents from the persistently depolarized end plate can cause repetitive, uncontrolled firing as the rest of the membrane recovers. The result is muscle spasm.
Okay, so mimics cause spasms by overstaying their welcome. What about drugs that mess with the cleanup crew, the acetylcholinesterase?
That's another major category. These drugs inactivate acetylcholinesterase. Common examples are neostigmine and physostigmine, which are used medically, but also some really nasty things like certain nerve gases, such as disopropyl fluorophosphate.
And if you stop the enzyme, acetylcholine builds up.
Exactly. With each nerve impulse, more ETSI is released, but it doesn't get broken down. It accumulates in the synaptic space. This means more receptors are activated for longer periods.
Leading to
More intense stimulation of the muscle fiber. Even a single nerve impulse can cause repetitive firing and severe muscle spasm, like the mimics, but often much more potent because you're amplifying the body's own signal. It can be lethal if it affects respiratory muscles, causing things like laryngeal spasm.
and the duration varies.
Yeah, neostigmine and physostigmine bind to the enzyme for several hours, so their effects are temporary. But nerve gases like disopropyl fluorophosphate can bind essentially permanently for weeks, making them incredibly dangerous.
Okay, so some drugs stimulate too much. Are there drugs that block the signal?
Yes, absolutely. The classic examples are the Curer-form drugs. The most famous is ditibocurin, originally derived from curare poison used on arrow tips. These are actually used medically sometimes, like during surgery, to cause muscle relaxation.
How do they block transmission?
They act as competitive antagonists. They basically sit on the acetylcholine receptors on the muscle fiber membrane, blocking the spot where acetylcholine would normally bind.
So even if the nerve releases acetylcholine, it can't find an open parking spot on the receptor.
Precisely. The curare-like drug occupies the binding site. As a result, acetylcholine can't bind effectively, the ion channels don't open sufficiently, and the end plate potential doesn't get large enough to reach threshold.
No action potential.
No action potential. The signal is blocked and the muscle can't contract. It causes paralysis.
This whole intricate system, being vulnerable to drugs, it also makes sense it could be affected by disease. Myasthenia gravis comes to mind.
Myasthenia gravis is a really clear example. Yes, it affects maybe 1 in 20,000 people, and the hallmark is muscle weakness and fatigue that gets worse with activity. It's a failure of signal transmission at the neuromuscular junction.
And what's going wrong there?
It's believed to be an autoimmune disease. The person's own immune system mistakenly creates antibodies that attack their own acetylcholine receptors on the muscle side of the junction.
So the body's destroying its own ears for the acetylcholine signal.
Essentially, yes. These antibodies can block the receptors, damage them, or make the muscle cell break them down faster. The end result is fewer functional acetylcholine receptors available at the motor end plate.
So even if the nerve releases a normal amount of acetylcholine,
There just aren't enough working receptors for it to bind to. This means the resulting end plate potential is much weaker than normal.
often too weak to hit that threshold.
Exactly. The weakened EPP frequently fails to depolarize the muscle membrane enough to open the voltage-gated sodium channels and trigger a full action potential. Signal transmission becomes unreliable, especially with repeated effort, leading to the characteristic muscle weakness.
And that weakness can be serious, right?
Oh, definitely. If it affects muscles involved in breathing or swallowing, it can be life-threatening.
Are there treatments that target this junction problem?
Yes. While they don't cure the underlying autoimmune attack, drugs that inhibit acetylcholinesterase, anticholinesterase drugs like neostigmine, can help manage the symptoms.
The same type of drug that can cause spasms if there's too much.
Yes, but used carefully here, the goal is to boost the signal. By slowing down the breakdown of acetylcholine, these drugs allow the ACE8 that is released to hang around longer in the cleft and accumulate to higher concentrations.
Giving it more chance to find the few working receptors.
Precisely. This increases the likelihood that enough receptors will be activated to generate an end plate potential strong enough to reach threshold and trigger a muscle action potential. Patients often get significant, though temporary, improvement in strength.
Okay, so let's assume the signal does successfully cross the junction. It generates an end plate potential, triggers that action potential on the muscle fiber surface. Now what? How does that electrical wave on the outside make the stuff inside the muscle fiber actually contract? The fiber is pretty big, relatively speaking.
Right. This is the next crucial step. Excitation-contraction coupling. Getting the signal from the surface membrane deep into the fiber's core, where the contractile machinery, the myofibrils, are located. An action potential just skimming the surface wouldn't work very well for a thick muscle fiber.
So how does the signal get deep inside?
Through a really amazing network of tunnels called the transverse tubules, or T-tubules. These aren't separate structures, they're actually deep invaginations, like fingers poking inwards, of the muscle fiber's own surface membrane, the sarcolemma. They tunnel right through the fiber from one side to the other.
So they're basically tubes of the outside membrane going deep inside
Hmm.
and filled with extracellular fluid.
Exactly. They carry the surface membrane potential and the action potential propagating along it, deep into the interior of the cell. These T-tubules branch and form an intricate network that runs alongside and wraps around all the individual myofibrils within the fiber.
Okay, T-tubules carry the electrical signal deep. What do they connect with or talk to down there?
They run in very close association with another specialized internal membrane system, the sarcoplasmic reticulum, or SR. You can think of the SR as a muscle cell's version of the endoplasmic reticulum, but highly specialized for storing and releasing calcium.
Ah, calcium. That's the key player inside, right?
The critical intracellular messenger for contraction, yes.
Huh.
The SR forms a lacy network of tubules surrounding each myofibril. It has these enlarged sacs called terminal cisternae that lie right up against the T-tubules, and then longer longitudinal tubules that run along the myofibrils. And its main job is to hoard calcium ions. It actively pumps calcium into itself, creating a very high concentration inside.
So electrical signal comes down the T-tubule. How does that tell the nearby SR to release its calcium hoard?
It's a direct physical coupling, essentially. As the action potential travels down the T-tubule membrane, the change in voltage is detected by special proteins embedded in the T-tubule membrane. These are called dihydropyridine receptors, or DHP receptors. They act as voltage sensors.
And these DHP receptors are linked somehow to the SR.
Yes, in skeletal muscle, they are physically linked across the tiny gap to another set of channels located in the membrane of the adjacent SR terminal cisterna. These SR channels are the calcium release channels, also known as ryanodine receptors, or RIR channels.
Physically linked, like touching.
It's believed to be a direct mechanical linkage in skeletal muscle. So when the action potential changes the shape of the voltage-sensing DHP receptor in the T-tubule,
It mechanically pulls open the ryanodine receptor channel in the SR.
that's the model. This opens the floodgates in the SR. The huge concentration of calcium stored inside the SR rushes out through these opened RAR channels into the sarcoplasm, that's the cytoplasm surrounding the myofibrils.
And it's this flood of calcium into the sarcoplasm that directly triggers the myofibrils to contract.
Bingo. Calcium ions bind to a protein called troponin, which is part of the thin filaments actin. This binding causes another protein, tropomyosin, to shift out of the way, uncovering binding sites on actin that myosin heads can then grab onto. That actin-myosin interaction is the molecular basis of muscle contraction. And those RIR channels only stay open for a few milliseconds after the action potential passes, delivering a rapid pulse of calcium.
Okay, calcium floods out, contraction starts. But for the muscle to relax again, you need to get rid of that calcium from the sarcoplasm, right?
Absolutely. Contraction will continue as long as the calcium level in the sarcoplasm is high enough to keep those troponin and tropomyosin switches turned on. Relaxation requires quickly pumping that calcium back out of the sarcoplasm.
And where does it get pumped? Back into the SR.
Exactly, back into storage. This is done by another protein machine embedded in the walls of the sarcoplasmic reticulum. It's a very active calcium pump called SERCA, which stands for Sarcoplasmic Reticulum K2 plus ATPase. It uses ATP energy to actively transport calcium ions from the low-concentration sarcoplasm back into the high-concentration environment inside the SR tubules.
It must be a powerful pump to work against that gradient.
Incredibly powerful and efficient, SERCA can concentrate calcium inside the SR tubules by about 10,000-fold compared to the resting level in the sarcoplasm. And to help the SR hold all that calcium, there's another protein inside called caliquestrin.
Caliquestrin, what does that do?
It acts like a calcium sponge inside the SR. It binds up lots and lots of calcium ions, maybe up to 40 per molecule. This allows the SR to store a huge total amount of calcium without the free calcium concentration inside the SR getting astronomically high, which would make the SERCA pump's job much harder.
So SERCA pumps it in, calsequestrin helps hold it, and this lowers the calcium level in the sarcoplasm, allowing relaxation.
Precisely. As sarcoplasmic calcium drops below the critical level, it detaches from troponin, tropomyosin slides back to cover the actin binding sites, myosin lets go, and the muscle fiber relaxes.
Can we put some numbers on these calcium levels? What's resting versus active?
Sure. In a resting muscle fiber, the free calcium concentration in the sarcoplasm is kept incredibly low, less than 10 arimolar. That's too low to trigger contraction. troponin and tropomyosin keeps things blocked.
And when the SR releases its calcium.
During full excitation, the calcium level in the sarcoplasm shoots up dramatically, potentially reaching as high as 2010 R-molar. That's a 500-fold increase. Interestingly, this peak level is actually about 10 times higher than the concentration needed for maximum muscle force generation.
Another safety factor, ensuring a strong response.
Seems like it, yeah, a robust signal. And then, almost immediately, those circopumps start working furiously to pump the calcium back into the SR.
So the whole spike in calcium is quite brief.
Very brief. The total duration of this elevated calcium pulse in a typical skeletal muscle fiber is only about 120th of a second. Contraction happens during that pulse. Of course, this duration can vary. Heart muscle, for example, has a much longer calcium pulse, maybe a third of a second, matching its longer action potential.
And for a sustained contraction, like holding something.
You need a train of action potentials coming down the nerve, firing repeatedly. Each action potential triggers a calcium pulse. If the pulses come close enough together, the calcium level in the sarcoplasm stays elevated, maintaining the contraction.
This whole calcium handling system, T-tubules, SR, DHP receptors, ryanodine receptors, sarcopumps, it's so tightly regulated. It makes sense that if something goes wrong here, it could be really bad.
Absolutely. And there's a condition that highlights this dramatically: malignant hyperthermia.
Malignant hyperthermia, what is that?
It's a rare but potentially fatal inherited disorder. It's triggered in susceptible individuals by exposure to certain specific agents, typically volatile inhaled anesthetics like halothane or isoflurane, or the muscle relaxant drug acetylcholine.
So it's a reaction to anesthesia. What goes wrong genetically?
Yes, it's genetic. Most commonly, it's caused by mutations in the gene for the ryanodine receptor, the RRR1 channel, in the sarcoplasmic reticulum. Sometimes mutations in the DHP receptor gene in the T-tubule are involved too.
And what do these mutations do to calcium release?
They essentially make the ryanodine receptor channel leaky or hypersensitive. So in response to these triggering agents, there's a massive, uncontrolled, unregulated flood of calcium out of the sarcoplasmic reticulum into the sarcoplasm. It's like the floodgates get stuck open.
Way too much calcium release.
Way too much, and it persists. This leads to sustained, forceful, body-wide muscle contraction and rigidity.
That must burn a huge amount of energy.
Exactly. The muscles go into hyperdrive metabolically. They burn through ATP like crazy, producing enormous amounts of heat. That's the hyperthermia, the dangerously high body temperature. It also leads to lactic acidosis, oxygen depletion. It's a full-blown metabolic crisis within the muscle cells.
What are the signs?
Severe muscle rigidity, incredibly high fever body temp can soar, rapid heart rate, and complications can be severe. Widespread muscle breakdown called rhabdomyolysis releases potassium and other cell contents into the blood, causing life-threatening hyperkalemia, hypotassium.
How do they treat such a dangerous reaction?
It's a medical emergency. Treatment involves immediately stopping the triggering anesthetic, aggressively cooling the patient down, and administering a specific drug called dantrolene.
Dantrolene, how does that help?
Dantrolene works by directly interfering with the ryanodine receptor channel. It inhibits that uncontrolled calcium release from the SR, helping to shut down the excessive muscle contraction and the runaway metabolism. It's often life-saving in these situations.
Wow. So we've really covered the whole journey today, haven't we? From that initial nerve impulse, starting way up in the spinal cord, traveling down the axon, triggering acetylcholine release at the neuromuscular junction.
Across that tiny synaptic cleft, activating receptors on the muscle fiber, generating the end plate potential, sparking the muscle action potential.
Which then dives deep into the muscle fiber via the T-tubules, talks to the sarcoplasmic reticulum through that DHP ryanodine receptor coupling.
Unleashing the calcium flood that activates the actin and myosin filaments, causing contraction. And then finally, the rapid pumping of calcium back into the SR by circa, allowing the muscle to relax.
And we've seen how fragile points in this pathway can be. The ACA receptors in myasthenia gravis, the calcium channels in malignant hyperthermia, and how drugs can specifically target these molecular players.
It really underscores the complexity. Aiming for that sort of elite professional level understanding, you really start to appreciate the incredible precision needed. The timing, the concentrations, the specific protein shapes and interactions all happening in milliseconds.
It's like a microscopic Rube Goldberg machine, but one that works flawlessly most of the time, billions of times over our lifetime. Just incredible biological engineering.
It really is. And understanding how it works normally is key to understanding what goes wrong in disease and how we might be able to fix it.
Absolutely. Which brings us to a final thought for you, our listener. We've dived deep into this one specific communication system, nerve to muscle. Considering the intricate molecular choreography we've uncovered, what does this tell us about other complex communication systems in the body? Think about synapses in the brain, or signaling within cells. How might similar principles of precise molecular interactions and perhaps similar vulnerabilities to disruption apply elsewhere?
And how might understanding these specific molecular defects, like a faulty receptor or a leaky channel, pave the way for developing more targeted therapies for a whole range of conditions beyond just muscle disorders? Lots to think about. Keep asking questions and keep exploring.